All right, guys, so we're going to be moving on to our next lesson, which is over the scientific method. And once again, a lot of this is going to be a review of things that you've covered in science before. But we need to make sure that we understand what is science, how does it work, and we need to understand some terminology related to experiments, because we're definitely going to have a lab component in this class. Remember, you have a lecture section and a lab section. It doesn't really feel separate to you guys, but in terms of your grades and in terms of at Texarkana College and getting credit, you have to kind of divide it into lecture and lab. So we have to make sure that we really focus on the lab components. And so this is going to be kind of the first portion of our lab is going over just some basic processes of science. And so what is science? You know, it's a subject that you've been studying since you were in elementary school. But the key is science is a division that investigates the natural world. And that's the key, is that it does have to be natural. Okay, so this is not going to be your supernatural. This is not going to be things like religion. We are specifically studying the natural world. It is very important that we be objective. And that means we don't put our own personal beliefs. We have to come in without bias. Okay, we have to let the evidence speak for itself, and we can't bring in any preconceived notions or what we think. It's not about what we think, it's about what the data shows. And so we have to be neutral as we study and do these processes of science. We also need to be methodical. Okay, that's why we call it the scientific method, and that there is kind of a method to the madness. There's a process that we follow. And it does have to be understandable and repeatable for others. Okay, it's very important that others can understand what we did and how we did it, and they have to be able to repeat it and confirm and hopefully show that our findings are valid. And so here we have the scientific method. It's not necessarily a 1, 2, 3, 4, 5 kind of thing, but it's essentially an ongoing process. Okay, there's a lot of different steps. Sometimes we have to backtrack and start over again. But once again, we see this word objective. It is an objective way to study the world around us, to make predictions, to test out our hypotheses, and to kind of determine if we've supported that or not supported that, and then go from there. So there are a lot of different steps, but as I mentioned, it is a process, okay? It is ongoing. It's constantly happening. You're doing science when you're standing in the aisle of Walmart trying to pick out the best product. If you're looking at deodorants or you're looking at hair products, anytime you are trying to answer questions and go through this process of what's going to work the best, those are the types of things that would fall under science. Even though you may not feel like a scientist when you're standing in Walmart, you are using some of these scientific processes. And so it typically begins just with a basic observation and a question. So it could be, you know, which one of these conditioners is going to make my hair the softest? Or if you know me, I have wild frizzy hair, which one of these products is going to tame the frizz the best? Okay, but it usually starts with some type of observation. Which one of these cleaners is going to remove that stain? Which one of these fertilizers is going to help my plants grow the best? We just usually start with a question of some type. Now, in order for it to be useful in science, it does have to be testable. And a good testable question is going to have two very important variables, the independent and the dependent. The independent is really what you are testing. What are you testing? Hair conditioners? Are you testing fertilizers? Am I testing music styles? So the independent variable is what you, the experimenter, is actually changing or manipulating. And then the dependent is just what happens as a result of that. Okay, the dependent depends on the independent. Sometimes these were called, maybe in younger grades, the independent was called the manipulated variable, and then the dependent is called the responding variable. And so you may that may be helpful to you if you know them that way. So you actually manipulate or change the independent, and then the dependent responds to that. You, as the experimenter, have zero control over the dependent. You don't get to control whether the stain actually comes out. That depends on the stain remover. You don't actually get to make the plant grow. That depends on the fertilizer. Now, once we create our question, this testable question, we're going to propose an answer to that question, and that is what a hypothesis is. and ES is just plural, okay? So a hypothesis is an educated guess or a proposed answer to the question. And it typically should always be in a format that contains both the independent and the dependent variable. There's not just one way to do it, but they should always contain both variables. And it's a statement, okay? It's not going to have a question mark. Let's see, I'm trying to think. Miracle-Gro will make my plants grow the most, okay? So my independent would be the fertilizer, so I picked a fertilizer. Miracle-Gro. Dependent was the plant growth. So when I say Miracle-Gro will make my plants grow the tallest, I now have both variables represented in my hypothesis. The key is it has to be testable. Some things we can't test. If it's based on opinion, we can't necessarily test that. Now, another thing that you might come across is something called a null hypothesis, and that just simply says that the independent has no effect on the dependent. You're going to practice writing these in a lesson. And so you could say fertilizers have no effect on plant growth. Stain removers have no effect on the removal of the stain. Conditioner has no effect on the frizziness of my hair, okay? So it's essentially just saying there's no relationship. That is also going to be testable because your test is either going to show that yes, there is a relationship or no, there is not. Now, another important thing for a hypothesis is you don't have to say why you think that, okay? Keep it simple, okay? It's a simple answer to your question. So you don't have to say, well, I think this product's going to work the best because. There is no because in a hypothesis. It is just a simple statement of what you think is going to happen in your experiment. And it is totally okay if your hypothesis ends up being wrong, okay? You do not have to be right. It just needs to be testable, okay? Now, in order to test this hypothesis, we're going to do a controlled experiment. And the key to controlled is that we should only test one thing at a time. I can't test water and soil and fertilizers and sunlight and then try to make a conclusion about plant growth, okay? I can only test just fertilizers, or I can only test just the amount of sunlight. I can't do all of them at once. So the one that you're testing is your independent variable. And in a controlled experiment, everything else is going to be kept constant. The only thing that we're changing or altering or manipulating is the independent variable, okay? So if I am going to test out fertilizers, my plants are going to have to get the same soil, the same amount of soil, the same type of pots, the same amount of light, the same amount of water. The only thing that I can alter is the independent variable. Otherwise, I don't know what led to my results, okay? Now, I will often also have a control group, okay? And this is going to be, in an experiment, you have your experimental groups, the ones that you are applying the independent variable to, and then you have a control group. And this is the one that does not get the independent variable. So if I'm going to test out different fertilizers, how do I know if the fertilizer was the only thing that affected the growth, okay? I need a control group that gets no fertilizer, because if all of my plants die and I don't have a control group, I can't conclude that fertilizer kills plants, okay? So I need that control group that got no fertilizer, because if I have a control group and all of my plants die, clearly it has nothing to do with the fertilizer, there was something else. But if I had a control group with no fertilizer and all the other plants got it and all the other plants died, then I could say that fertilizer killed my plants. So you need this comparison group that does not get the independent variable, okay? And that way we know that we're testing what we think we're testing, and when we get our results, they have some validity to them. So here's an example we have, in a controlled experiment. Notice in this particular experiment, we're testing water versus no water. So your control group got none, your experimental group got water. Notice there's the same number of beans, they're the same type of seeds, the same pots, the same soil. Here we have our control group for comparison, and then we look at the outcome of our plant growth. Now, sometimes we can't necessarily do a controlled experiment because when we're dealing with science and we're dealing with nature, if we're out in nature, it's really hard to control nature. We can't control things like the weather. We can't determine the wind speed. We can't keep the temperature constant if we are outside in a natural environment. So there are times when our experiments may not be perfectly controlled. And in that case, we do still try to have to be as consistent as possible, but it's going to be much more difficult to determine our results because there could be many variables at work. Okay, so if anytime we're studying like a natural ecosystem or we are out actually in nature, we are going to have to kind of be a little more careful. We can try to be controlled, but there's no way to perfectly control an experiment that is done in a natural environment. All right, so in a controlled experiment or in an experiment, we do need to collect data. Now, it's typically best if this data is quantitative. There's two general types of data. We have qualitative, which describes the quality of something. The tree bark was rough, okay? That would be a qualitative piece of information. The earthworm was slimy. The earthworm was skinny, okay? Those are all qualitative descriptions. Now, quantitative is typically better in science. If you can collect quantitative, this is what we want because this is where we have distinct numerical data. So if I say that the earthworm is skinny or the earthworm is short, those words can be different things to different people. But if I say the earthworm is 25 centimeters long, 25 centimeters should be the same to everyone. Okay, so quantitative is going to be better. It's hot outside. Well, hot to me could be 95 degrees. Hot to you could be 85 degrees. So it's important that we use quantitative rather than just using descriptive words if possible. There's a place for qualitative data, but if we can apply numbers, we want to apply numbers because that way they are distinct and easy to compare and we know exactly what they are and they mean the same to everybody. And in science, by the way, we do use the metric system, okay? We do not use, we wouldn't use 85 and 95 degrees. We would use the Celsius temperature scale. Now, your collection should be consistent. We do need to be careful with taking measurements. If you have a sample size, you should always do larger is better. And here's the thing, a lot of times we want to be right, and so sometimes we want to change the data to fit what we think should happen. Never do that. The data is what the data is, and we have to go with it, even if it's unexpected. Okay, we can't change it. Now, once we get our data, we have to interpret those results. In science, depending on your experiment, sometimes it could be inductive, where we're going from specific to general, or it could be deductive, where we're going from general to specific. Depending on your scientific experiment, we use both in science. But essentially, you take the data that you've collected and you have to make some conclusions. So like here's a lot of times inductive reasoning. If you cough when you eat peanuts, you must be allergic to peanuts, okay? So we take, essentially we're going from specific and drawing a general conclusion. Deductive is going the opposite way, okay? We go very specific, or we get specific from general information. All right, the last thing we're going to talk about, well, not the last thing actually, is graphing our data. And you are going to be practicing doing some graphs tomorrow in class. So graphing data is really important for kind of displaying our results. Now, anytime you do a graph in science, you want to make sure that you have the good or the elements of a proper good graph, okay? So every graph should always have an appropriate title. This usually includes the independent and dependent variable. You must label the X and Y axis. The independent typically goes on the X and the dependent typically goes on the Y. You do need to include units if you have measurements like time. Is it minutes? Is it seconds? Is it hours? Is it days? You need to use regular intervals and even increments. So like I need to count by twos, or I need to count by tens, or I need to evenly space my bars. If you have a line graph, you can connect the plotted points. If you're going beyond your graph, you need to use dotted because that's not actual data. And when necessary, you should always have a key for your graph. And so here's an example of some elements of a good graph, okay? Anytime you do graphs in your labs, they will be graded, and these are the things that we will be looking for, okay? Now, how do you know if you do a line versus a bar graph? Well, a line is typically change over time. If it's continuous, like growth over a period of time, that would be a line graph. Bar graphs are typically categorical. They're independent categories that don't really necessarily affect each other, like colors or groups in school or something like that. Now, once we have collected our data, we analyze our data, now we have to draw conclusions. Now, if we're doing one experiment, we're not proving anything. We're either supporting or not supporting our hypothesis. So we really shouldn't be using the word prove. This is why in science we must always repeat to ensure that those results are getting replicated over and over and over again. Now, once we have our conclusions, especially if we have something that's pretty significant, we need to get that information out into the public. And so it is important that we publish our findings. Now, if we do an experiment in dual credit biology, we're not going to be publishing anything. But if you were doing research for a college or you were a researcher for your job, once you established these findings, you would have to publish them and they would undergo the peer review process. And this is where experts, people in that field, are going to test, they're going to retest, they're going to ensure that everything was done properly and they are getting similar results, okay? And we also want to publish to get that knowledge out there so that other people can build on that information. Now, once we get information that is shown to be true, it's been peer reviewed, it's been shown to be true over and over and over again, we can take an idea that was a hypothesis and eventually it can become a theory. Now, a theory is not weak. It takes a lot to become a theory. They are very well established, they are reliable, they are widely supported by the scientific community, they come from repeated testing by multiple people, and so they're not weak, okay? That doesn't mean they're set in stone, okay? With more information, they could be changed, further supported, or possibly thrown out. But a theory to reach that level is a very strong piece of information and evidence, and it is something that has been shown to be true many times, okay? Now, something that's even more solid than a theory is a scientific law. And the thing with a law is that there's never been exceptions. As long as you drop something, it's going to go down. Gravity's going to pull it down, okay? So when it comes to your scientific laws, these are observations that are pretty much universally understood without exceptions. And we don't have to understand why it happens. We just have to know that it does. Okay, Newton's laws, the law of gravity. So there's a lot more, probably a lot more in physics than there are biology. But just know that laws are about as solid as you can get. And that is going to wrap up our lesson on the scientific method. You are going to have some practice coming up to help make sure that you understand these concepts. And just like the last set of notes, the Intro to DC Bio, these will also be on your quiz that we are taking over this kind of first unit. So I hope you guys have a great day, and I hope to see you soon.